Hello and welcome. It's time for Asynchronous Lecture 21 for Bio 145 Microbiology. This lecture is scheduled for Monday, March 16th, 2016, and this is our third and final installment on the topic of antibiotics. Let's do a little bit of a recap. First, a little recap. We're talking about 4 ways a microbe can become resistant to an antibiotic. So we think of a mutation in a gene that causes a new protein activity, and sometimes it can be a different rRNA product, but it's usually protein changes. Okay, so one example would be a mutation in a gene that encodes a protein that is involved in the entrance of the antibiotic. So let's say if we're gram negative, the antibiotic has to go through a porin in the outer membrane of the cell wall and the plasma membrane. It's got to go through maybe some type of channel there. So a mutation in those kinds of genes, porin, channel, could lead to antibiotic resistance because of the mutation, the antibiotic can't get in. Let's say, for instance, if it's going in the cytoplasm, it can't go through the porin or it can't go through the plasma membrane channel. A second possibility would be a mutation in a pump. So a pump protein or translocase. So because of the mutation, the protein changes. So instead of just pumping things like sugar or iron or some metal, it starts to pump the antibiotic out of the cytoplasm and out of the cell. Okay. Third possibility would be there's a change in a gene that encodes a protein, and now that protein, using enzyme, it goes from doing what it normally does to inactivating, breaking down an antibiotic, usually through some chemical modification. So the enzyme, instead of breaking down sugars, it starts to break down an antibiotic, or it phosphorylates an antibiotic, or does something like that to inactivate it. And the last one could be that the antibiotic has a binding site in the cell. Maybe it's a protein, or maybe it's an rRNA if it's in a ribosome. A mutation in the gene changes the protein that the antibiotic is binding to, or the rRNA that the antibiotic is binding to. Okay? Because some antibiotics bind to other things other than proteins. All right, so those are like 4 types of, gene targets that when they get mutated can lead to antibiotic resistance. So the next thing is, well, how do you measure antibiotic sensitivity or resistance? So it's what's usually used is called a Kirby-Bauer disc assay. It's a very simple assay. It's pretty easy to understand just because you just look at things, all right? And you see the difference of a diameter of something, okay? So what happens is you take a lawn of bacteria and you spread them out on a nutrient auger. You just spread it out. Now you don't let them grow yet, okay? So right after you spread them out, you apply discs that contain different antibiotics. Okay, these are like little paper discs, and they're kind of soaked up the antibiotic, and then they're dropped carefully on this plate that has this lawn of bacteria that hasn't grown yet. Then you allow them to incubate, usually overnight, and the next day you look at the discs and you see how close the bacteria are to the discs. If they've grown up to the edge of the disc, that means the antibiotic doesn't do anything to them. But if there's a large area where there's no growth, we call it an area of inhibition around the disc that had the antibiotic. Then you say, you can see, I can see that the bacteria has killed what would have been, the antibiotic has killed the bacteria that should have grown in this area. So we call it a zone of inhibition because the bacterial growth has been inhibited around this area of the antibiotic. So the more resistant a bacteria comes to an antibiotic, the smaller that zone, the smaller the diameter. All right. So the diameter is a measure of sensitivity. So let's look at the next illustration to kind of clarify this. For the Kirby-Bauer disk assay. So let's just start over here with 1B. Again, you've spread the bacteria out on this nutrient agar plate, and then you apply these little antibiotic discs. So these are each a different antibiotic. Okay. You put them, touch it gently, make sure they stick there. And then you grow it overnight, and you look at the growth of the bacteria. And you can see kind of here, you can see with the brown kind of lines, or you can see, oh, they're growing. And then there's these clear patches, okay? So from, you can see from the edge of the antibiotic disc to the edge of growth. And in this zone of inhibition, here's your zone of inhibition, the bacteria have not grown because of the antibiotic diffusing out and killing it in this area. So they all been killed in this area. All right. And if you want to put a number on it, you can actually measure the diameter. So the larger the diameter from here to here, that means the greater the zone of inhibition, the more the bacteria are sensitive to that antibiotic. On the other hand, here you can see this one, the zone of inhibition is very small. The diameter would be very small. So you'd say, oh, these bacteria seem pretty resistant to this antibiotic. And this one has no zone. So you say that these are absolutely resistant. Okay? So that's how you can measure sensitivity or resistance. So let's take a question now, kind of put the stuff from the previous lecture and today together. You have a gram-positive bacterium. It's become resistant to cephalexin. You suspect the mutation has occurred in. Okay. So thinking about the 4 types of genes that are targeted, that mutations can lead to resistance. Yeah, think about that. You think of cephalexin. Okay, cephalexin. That's a cephalosporin. Okay. That inhibits transpeptidases. So why don't you pause it now and think about these answers, and I'll give you the answer. Okay. Okay, here's the answer. So first of all, what would lead to, well, would it be, yeah, it would be a transpeptidase, definitely, because cephalosporins inhibit transpeptidases. 30S ribo, no. Cephalosporins don't affect translation. A plasma membrane pump. Now, a lot of people might jump and say, oh, yeah, something's going to pump the antibiotic cephalin out of the cytoplasm and out of the outside the plasma membrane. But folks, remember, cephalexins, they're effective outside the plasma membrane. Their target is that area of cell wall synthesis outside the plasma membrane. So a plasma membrane pump wouldn't have anything to do with it. So that's not right. That's not right. So the answer is A, transpeptidase. Okay. So just remember location. Where does the antibiotic need to work? If it needs to work in the cytoplasm, then yes, a plasma membrane pump would, if it's mutated, would make that cell antibiotic resistant. But if the antibiotic works outside the plasma membrane pump isn't going to affect resistance or sensitivity. Think location, 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 location. Just like if you're going to set up a store in a town. So some antibiotics, are rendered useless by certain bacteria because the bacteria have become resistant. But what researchers have found is sometimes you can couple an antibiotic with some partner, and the partner can inhibit that enzyme or whatever the bacteria makes that makes it resistant. And therefore, if you can inhibit that, then the antibiotic will work. So we say some antibiotics are given a partner, and the partner is the inhibitor of the resistance protein. So one example I'd like you to know is augmentin. And augmentin is, you might have been treated with it. It's a combination of amoxicillin, which is a penicillin, and clavulanic acid. Clavulanic, or clav, it's known as. Okay? So clavulanic acid is the partner. It's the friend. It has no antimicrobial activity by itself. So if you give bacteria clavulanic acid, it doesn't hurt them at all. But what it does do when it's coupled with penicillin, it's a very powerful helper. It is an irreversible inhibitor to the binding site of beta-lactamases. So you can imagine if a beta-lactamase is going to destroy a penicillin, well, a penicillin's useless. But if clavulanic acid's around, clavulanic acid binds the beta-lactamase, stops it so it can't destroy the penicillin, and now the penicillin can work. So we kind of think of it's like almost the clavulanic acid is like a decoy to the beta-lactamase. It soaks up the beta-lactamase so it doesn't pay attention to the penicillin. And in stopping the penicillin from being broken down, now the penicillin can go and do its job on the transpeptidase and inhibit cell wall synthesis and end up killing the bacterium. All right, so the bacterium might have been originally resistant to penicillin because of its beta-lactamase, but clavulanic acid kind of nullifies the beta-lactamase. Now that doesn't work on all beta-lactamases, but it does on some types of beta-lactamases from some resistant cells. So a couple thoughts on resistance. First of all, cross resistance means if a bacteria is resistant to one antibiotic, it might be resistant to several structurally related antibiotics. All right. So, for instance, if a bacterium is resistant to penicillin G, it has this beta-lactam nucleus, well, that bacteria might also be resistant to some other penicillins like methicillin and cloxacillin. All right. Now you could argue, well, maybe the side chain here would, maybe that would stop the beta-lactamase from accessing the beta-lactamase. And that's true. That's true. But I'm just saying that's something you have to keep in mind. Cross-resistance is a possibility. So if a bacterium is resistant to one member of the tetracycline family, it might be resistant to many members of the tetracycline family. If it's resistant to one macrolide, it might be resistant to many, many macrolides. So you got to keep that in mind. Okay. Secondly, resistance is a localized phenomena. So it's not like if you went throughout the United States and you say, well, I found some bacteria that are resistant to penicillin G in California, then there must be in every state in the Union. No, it's not true. Resistance is a localized phenomenon. Even from close places like in Fresno versus Stockton, you might find in Fresno bacteria that are a lot of bacteria that are resistant to a given antibiotic. But in Stockton, there wouldn't be that many bacteria that are resistant to that same antibiotic. Same is true in rich versus poor hospitals in the same city. Okay? In the rich hospital, I bet you'll find a lot more bacteria that are resistant to certain antibiotics than a poor hospital. So actually the poor hospital is actually better in terms of not having resistant bacteria. Why? Well, here's where there's a high usage of an antibiotic, there's going to be bacterial resistance. So it's like if an antibiotic is used a lot in a certain hospital, in a certain town, certain state, you can pretty much guarantee there's a pretty high likelihood there's a lot of resistant bacteria resistant to that antibiotic. But if that antibiotic for some reason is not used in that hospital, not used in that city or the state, then probably there's not as many resistant bacteria to that antibiotic. Why is that so? Well, now we're going to go to the next idea about how resistance becomes widespread. Okay? How does resistance become widespread in an area or in a hospital? How does it become common? How did the bacteria become common? So first of all, it's a four-part process. How does resistance become widespread? Meaning, how does there's suddenly a lot of bacteria around that are resistant to a given antibiotic, versus maybe earlier there were very few that were resistant to that antibiotic. How did it become so widespread? Four-step process. The first step we've already talked about. There's a mutation. It creates a resistance factor gene, and it's totally random. So antibiotics do not cause resistance to pop up. Bacteria just randomly undergo mutations, and by luck, a mutation will lead to resistance. Okay, so think, okay, mutation in a porin, mutation in a pump, mutation in an enzyme that leads to degradation of the antibiotic, mutation in the binding site. Those four mechanisms. And like I said, you don't need the antibiotic to trigger the mutation. Mutations happen all the time. For instance, if you went back to Arizona in a time machine, 1852, would you find an antibiotic resistant bacterium, bacterium that's resistant to the antibiotic daptomycin? That's something that came up about 10 years ago. It's a fairly new antibiotic. But if you went back in time in 1852 and you looked for the bacteria, would they be resistant? Could you find one that's resistant to daptomycin? And the answer is yes. Yeah, back then bacteria have been mutating since the beginning of time, and by luck you could find a bacterium that if you applied came from the future and you applied daptomycin, yeah, you would say, yeah, that bacterium is resistant to daptomycin. Well, how can that be? Because mutations are random. They're completely random. Okay, the antibiotic presence does not cause the mutation. So, for instance, a missense mutation converts a normal pump protein into a multi-drug resistant pump protein. All right? And you could say this would explain why a bacterium is resistant to macrolides. Okay? So let's kind of keep that example as we go through this. So the second step is really important, and that is selection in the environment. Okay? There's got to be selection in the environment for those bacteria that are resistant to the antibiotic. Right? That's key to them becoming widespread. Why? Well, if the antibiotic is around, so we have our macrolide resistant bacteria. If the macrolide's around, all right, now that bacterium with that mutation that makes the antibiotic resistant has an advantage over the ones that are sensitive. And that one that's resistant, okay, because that bacterium is around, because that antibiotic is around, is going to outcompete the non-mutant cells. The non-mutant cells are all going to die off. Okay? And now there's no competition for the resistant mutant cells. So the resistant mutant cells, they have no competition, and they just grow and they take over everything. And now they become the dominant species in that population. This because the sensitive population isn't around to compete against it. It takes over. That's all that's left. There's no competition. It's just like if you were just to suddenly wake up in the morning and you had grown a pair of snowshoes, all right? And you looked out and there was snow all around. And everybody else around you did not have these snowshoes on their feet. Well, because you had snowshoes, you'd be able to go out in the snow, get food, get water, do all these things. You'd be able to have children reproduce. While those people who weren't lucky enough to have been born with snowshoe feet, they would die off. Because they can't compete against you. You, and therefore your generations of children, take over. Because you have this new ability. Snowshoe feet. Okay? However, what if the antibiotic isn't around in the environment? So there's a random mutation. The bacterium is resistant to macrolides now because of this pump. What's going to happen? Well, folks, now that mutation is a disadvantage. If there's no antibiotic around, what's happened? Well, that cell has a different pump, but probably that pump doesn't work as well. All right? It's supposed to maybe pump, like I said, what did we say? It was supposed to be pumping out originally it was pumping out, let's say, sugars or ions or metal ions. Well, now it pumps out an antibiotic, but it doesn't pump out those sugars or iron as well. So now it can't compete with those non-mutant bacteria. The non-mutant sensitive bacteria, their pumps work better than the antibiotic resistant pump. All right? The bacteria that are resistant to the antibiotic, though, they have a disadvantage now. There's no antibiotic around to select for them. So basically, they're competing against the antibiotic sensitive bacteria, and they don't have as good of a pump. And there's no antibiotic around. So guess what? They're going to die off pretty quickly. And the non-mutated sensitive ones, they're going to be fine because there's no antibiotic around. So you get the idea that if you do have a resistant bacteria, if the antibiotic isn't around, they're actually at a disadvantage and they're going to die off pretty quickly. So the antibiotic has to be around to select for the resistant bacterium. Okay? So like if you were born and you wake up and you got snowshoe feet, go out there, but there's no snow, you're going to have a hard time getting around those snowshoe feet. Trust me. The people without the snowshoe feet are going to be faster, quicker, get the food, get the water. And guess what? You're going to die off. Okay? So when you say selection now, what do you mean selection in the real world, not snowshoe feet, Jeff? Well, selection for resistant bacteria is, well, the presence of the antibiotic. But it's usually the presence of the antibiotic due to indiscriminate use, meaning the antibiotic is around in the environment when it shouldn't be. People are using the antibiotic excessively for unnecessary ways. So because of that, there's a lot of antibiotic around, and therefore that acts as a selective for the resistant bacteria, and that will make them become dominant because it'll kill off the sensitive strains, but these ones will now overgrow. So what is indiscriminate use specifically? There's three classic examples. Number one, prescription of an antibiotic for a viral infection. So humans, they basically expose bacteria. They select for bacteria that are antibiotic resistant in their body by basically taking antibiotic when they shouldn't. The classic is they have a cold, which is caused by a virus. They go to the doctor and they get an antibiotic. And that antibiotic is for bacteria. It's not going to help fight the virus. But people want the antibiotic. They want something. My friend, his name is Rob. I won't give his last name. He went to a reputable medical school. He is a doctor in Wisconsin. And he is a good guy, and he's a moral guy, but he hates confrontation ever since he was a buddy of mine in college. And he will try to explain to, for instance, parents that their children have a cold, and the parents just will not go away. They say, I want an antibiotic, I want it back. And after a while, he'll just say, I give up. I don't want to fight. And he goes, just to get rid of him, I'll give him a prescription for antibiotic, even though I know it's not necessary, it's not needed, it's indiscriminate use. That's one example. Another example is using antibiotics at low levels to prevent infections. All right. And the problem is it doesn't, when you use it at a low level like that, all right, and you're using when, there's no disease, yeah, it'll prevent it first, but that because they're being used at a low level, bacteria don't need that many mutations to suddenly become resistant to it. All right. And this is used a lot. Some people do use antibiotics to basically prevent disease, even though they're perfectly healthy. One example was, this was documented as a classic example of widespread penicillin resistance, was in the Philippines. There was a house of prostitution next to a military base. The military men would go over to the house of prostitution for services, and women would come down with, for instance, gonorrhea. And when they would come down with gonorrhea, they obviously couldn't partake in the business. Well, the people who ran the house of prostitution figured, hey, why don't we start giving out penicillin every day to these ladies, and that'll prevent them from getting gonorrhea. And that would work for a while, but then the bacteria would become resistant at that low level. So they'd have to increase the level of penicillin the ladies were taking. And soon the bacteria, you just need another mutation. Boom, they would jump. They become resistant to that level until finally They were completely resistant to any level of antibiotic the women would take. So low use of antibiotics for prevent, again, that's example of indiscriminate use. It doesn't kill the bacteria efficiently. It makes it a lot easier for the bacteria to become resistant to it, an antibiotic. And the third that's really bad in this country is the inclusion of antibiotics in animal feed. Animal feed, yeah, like for livestock, cows, pigs, chickens. Two things: disease prevention and growth enhancement. So, yes, farmers do play a role in antibiotic resistance. They provide the selection for the resistant bacteria. Here's an example. This is from the National Research Council. Tetracycline, or a combination of oxytetracycline, that's a next generation, and neomycin, you can look at the amounts, are used in starter feeds for turkeys and broilers, chickens for food. It's normal, it's allowed, all right? And you think, well, it's only a few hundred grams per ton. Well, how many tons of chicken food are used in the United States every year? Or turkey food? It adds up. It's a lot of antibiotic exposure. If you don't believe how easy it is to get, go to this website. This is a site where you can buy antibiotics for animals. And it's amazing the incredible array of different antibiotics you can get for your animals. All right? For farmers, go to the site and buy pounds of it, of antibiotic. It's much easier to get antibiotics for farm animals for people. It's crazy. So you might talk to a farmer now. Here's Farmer Bob. There's Farmer Bob. And he says, well, number one, he says the specific antibiotic in my food, it's not used on people. It's just used on livestock on my farm. Well, why is it bad? It's not getting to people. It's just on my farm animals. And he goes, and two, you look at the that website that sells the antibiotics. A lot of them are like first generation antibiotics. And he'll say, I'm just giving them tetracycline. That's not used on people anymore. So why do you worry? Why do you worry about these things? Number one, okay, can the antibiotic get from the farm to the person? So here is our little sketch drawing here. And you can imagine that the antibiotic is given to animals for disease prevention, control, growth promotion. There we go. Antibiotic kills the susceptible ones. And now you get resistant bacteria that grow on these animals, all right, on their skin, things like that. Well, so the bacteria, these resistant bacteria on the animals. Well, guess what? Those resistant bacteria spread to other animals. Okay. The resistant bacteria then spread to consumers, people, because they get on the meat. Okay, let's say if they're on the skin, they get on the meat's packaged. You get to people, you touch, raw meat, raw chicken when you're preparing it. Guess what? You rub it on your skin. Even if you cook it really well and kill the bacteria, you can still rub it on your skin, rub in your respiratory tract. Guess what? You're infected. Also, the bacteria from the farm get to farm workers. And hey, they're going to come to town and you're going to meet them. You're going to talk to them. You're going to shake their hands. Boom, you're going to get them. Also, that resistant bacteria can get to soil and water. Okay, that soil and water then it's used in irrigation, gets on fish, fruits, veggies, consumed by people, touched by people. So it does get to you. Those resistant bacteria are going to get to you many different ways. But then you're like, oh, hold it. But so what? Those bacteria are resistant to a first generation tetracycline. Okay, we don't use first generation tetracyclines. We're using third or fourth generation tetracyclines. So why should we worry about those bacteria on us that are resistant to tetracycline? Because of, what's that term we talk? Cross resistance, right? Those bacteria that are resistant to tetracycline that you've now been infected with, okay, they're resistant to, but they might also be resistant to second, third, fourth generation tetracyclines, the ones that they're used to treat you right now. So, Farmer Bob, those aren't two very good reasons at all. the microbiologist just shot them down. All right. So we're in the middle or the end of the second step. We had number one, some type of mutation. Number two, selection for bacteria that have that antibiotic resistance mutation. Mainly exposure, that bacteria are exposed to it. So the ones that are resistant survive and those ones that don't die. All right. I want to note that within this step, there can be multiple mutations that accumulate within the same bacterial cell or a bacterial like generation or population. So for instance, you could say that we have a bacteria first, there's an MDR pump mutation. And that happens in a bacterium. And then over a little time, then there's a mutation in a degradative enzyme. And then finally in a mutant target protein. Okay, there's a mutation in the target protein where the antibiotic usually binds. And let's say this doesn't happen in the same cell, but happens once, one generation, and then the second generation, the degradative enzyme, and the third generation, there's the mutant target protein. Now, so what happens is the mutations are accumulating, we'd say, in that strain. And they're kept around because there's always that exposure to the antibiotic. Okay. Now, there's a really cool video on YouTube. There's the link. It's not that long, and it just illustrates this point where they're going to start some bacteria on this huge like table that's made of agarose media with nutrient. And they start the bacteria in very low amounts of antibiotic, and they stop growing there until a mutation happens, and then they can start growing again, and they grow in higher levels of antibiotic, and they stop, and then mutation happens again, and now suddenly they become resistant to that higher level, and you see them basically these bacteria mutating and becoming resistant to more higher and higher levels of antibiotic. And what's seen on this huge basically table of media is actually kind of what's happening in society today. It's a really neat video, and it kind of brings this point out. Okay, so now you say, okay, I've got, let's say, a strain of bacteria, a bunch of them, and they have maybe one or several mutations that make them resistant, and those have been selected for. So there's a lot of those bacteria around, but it's not widespread yet. We're still only in maybe one species. How does it get widespread? Well, as you know, these mutant resistance genes now can be shared, okay, through recombination. So they go from one species to another by, for instance, transformation, specialized transduction, generalized transduction. So now these antibiotic resistance genes are in multiple, lots of different species. So that's not the end of it though, because remember, just because a bacterium picks up a gene doesn't mean it wants to keep it around. New genes, remember, like plasmids, are kind of burdens. They have to pay their way. They have to be worth it for the bacteria to keep them around. So why would it be worth it for a bacterium that picked up an antibiotic resistance gene? Why would it be worth it to keep it around? Well, if there is antibiotics in the environment, right? So along with step 3 has to come step 4, meaning there's selection. There's exposure of those bacteria that just picked up the antibiotic resistance gene. They're now exposed to antibiotics in the environment, i.e. those bacteria are on people that are using the antibiotic. So think there's more indiscriminate use, all right? That selects for the maintenance, the keeping of these antibiotic resistance gene in these bacteria that just picked them up by recombination. All right, and last you want to think of that steps 3 and 4 can happen multiple times, all right? So that you can have one strain of bacteria, many generations, but the same strain, and they're accumulating more and more antibiotic resistance genes, and those will be selected for by higher and higher levels of antibiotics in the environment. So that kind of gives you an idea of the 4 steps that lead to widespread, think multiple species resistance to an antibiotic. Okay. Let's go to a question now. Dr. Rob U. Blind has prescribed that his patient take a broad spectrum antibiotic for 1 month. Broad spectrum for 1 month, quite a while. You believe that? Okay, so he may not know the exact pathogen. She may not suffer from an overgrowth of Candida albicans. increase the chance of resistant bacteria developing compared if she'd only taken it for 10 days? A and C, all the above. So you should take a break right now and stop it and think and figure out the answer. And I'll tell you the answer. Okay? The answer, don't peek. Okay. The answer is all the above. Okay, why? Number one, broad spectrum is often used when the doctor, he or she, does not know the exact cause. So that's true. She may, okay, well, remember, if you take an antibiotic, a broad spectrum for a long time, all right, it'll wipe out a lot of the sensitive normal flora. But this Candida albicans, remember I kind of mentioned that in the asynchronous lecture. This is actually a fungus. It's resistant. Okay, it's not going to be hurt by antibacterial antibiotics. It's resistant to antibiotics. So, and it's normally in the vagina. And now, if all the bacterial competition is gone, this fungus is going to overgrow, and we get a super infection. Okay, remember, super infection. It becomes an opportunistic pathogen. Remember that too? Opportunistic pathogen now, because it's a super infection. Opportunistic, and that's when you think of a yeast infection. Okay. And so that's correct. And then the third one is this may increase the chance of resistant. Yeah, because you're having a person use it for 30 days, not 10 days. That's a longer time for selection. So that increases the chance that the resistant bacteria developing compared to that for 10 days. Yeah, becoming widespread. Okay, sounds good. Sounds good. All righty. So let's kind of go into some concerning issues with antibiotic resistance. And I think you might have heard of these three examples, but I just kind of cover them now. And when we get to the diseases, I'll refer back to them. So number one, you've probably heard of MRSA, methicillin-resistant Staphylococcus aureus. So Staphylococcus aureus is a species of gram-positive, okay, bacteria, and it's found a lot in a lot of people's normal flora. You're going to find out, okay? It's very common. So when you first of all you think, okay, it's a gram-positive bacteria, methicillin. Methicillin is just a semi-synthetic penicillin. So originally they found, oh, there's a lot of Staphylococcus that are methicillin resistant. But what they found in this group is they was actually resistant to many penicillins, not just methicillin. And the common denominator was that this MRSA, these methicillin-resistant Staphylococcus aureus, they have somehow acquired a mutant transpeptidase, all right, through some recombination event, and it spread very, very like wildfire among the Staphylococcus aureus. So now they all have these mutant transpeptidases that have a very low affinity for methicillin and other beta-lactams, meaning other penicillins. So because of their mutant transpeptidases, they just aren't bound by methicillin and other penicillins, and therefore they're resistant. Okay. Now, these bacteria are often on the skin. They're spread through contact, touching. They can live on fomites for quite a while. Weight rooms, really bad places. They live on the fomite, and they get picked up from someone who sits on the bench after a person who has a reservoir sat on the bench. That's why you should wipe it down with those disinfectant wipes with the quats in them. Okay, so and you think, well, so I picked up some Staphylococcus aureus. Well, they can cause skin infections, and some of these can be pretty bad skin infections, you can see. And depending on the strain, some of these bacteria some of these MRSA can actually get into the blood and lead to septicemia and even be life-threatening. Okay, so MRSA is not something to be taken lightly. Here's an interesting fact. 1/3 of people that were tested in the U.S. have S. aureus in their normal flora. So it's 1 out of every 3. Okay, so in our class of 150, that means about 50 of you have Staphylococcus aureus in your normal flora, usually the skin, and 5% of those strains are MRSA. Okay, 5%. So 5% of 50, that's 2.5 people. Okay, in my 150 students. Okay, think about that.
So MRSA is a major concern. But even a greater concern might be gram-negative bacteria, a lot of different species, that have been found to contain extended spectrum beta-lactamases, or ESBLs. Just a reminder from a previous slide the other day, beta-lactamase, BL, that's an enzyme that degrades beta-lactam rings. Beta-lactam rings, remember that's like the house with the little chimney. They're found on things like penicillins and cephalosporins. All right, but an ESBL, this is the extended spectrum part. Okay, we're talking these ESBLs. These are proteins, right? They're enzymes coded by genes. They can attack and they can break down all kinds of penicillins, all cephalosporins. Monobactam is another group of antibiotics. We don't talk about them a lot in this class, but they're out there. And they can even break down certain inhibitors like clavulanic acid. All right. So instead of like sticking in a beta lactamase and clogging it up, that inhibitor, no, these beta lactamases can actually chew those things up, which are pretty amazing. All right. So if a bacterium has this, and usually these are found in gram negatives, so if the gram negative has this, that gives them the potential to be resistant to a lot of different antibiotics. Now, well, it's like, well, is it easy for a gram negative bacterium to get a gene like this? And unfortunately, yes. So these genes that encode the ESBLs, they're often found in a lot of different mobile plasmids out there. All right, they're circulating around. And on these plasmids is not just the extended spectrum beta lactamase, but there's amazingly, they're also found on plasmids with resistance genes to other kind of things like fluoroquinolones and aminoglycosides. So you can imagine if a bacteria picks up a plasmid and it gets an ESBL and it gets a resistance gene to fluoroquinolone and something else, oh my gosh, that bacterium becomes like a super bug. It's very hard to kill. How would it get them? Well, just thinking of plasmids alone, okay, plasmids, we think about transformation, all right, uptake of naked DNA, and also, what way a plasmid, a whole plasmid, you can write this in yourself, conjugation. So there you see the evil of conjugation. Remember with the conjugation bridge and the pilus and all that stuff. So unfortunately, these are spreading to more and more bacteria over time, which gives them these resistance. Okay, well, not done yet though. Keep going. And the third big concern is antibiotic resistance that's found in certain strains of Mycobacterium tuberculosis. So remember, for treatment of Mycobacterium tuberculosis, all right, people take several antibiotics, and they have to take them for months. Okay, kind of talked about that earlier. Now, when you think about it, when they're taking them for months, and they're taking them, those Mycobacterium tuberculosis in the person's body, they're going to go under spontaneous mutation, and then since the antibiotics around, they're going to be selected for. So you're going to see that develop within the person's body often. So for instance, one group that you have to worry about are the multi-drug resistant Mycobacterium tuberculosis. That's MDR-TB. These are strains, and these are usually resistant to two classes of antibiotics. What they are is usually to rifampin and isoniazid. Because why? Because those are the two classes that are usually used first on a person when they are diagnosed with tuberculosis. All right? These are known as the first-line drugs. You take them for a long, long time, they're going to select for resistant, and there's a chance that those bacteria during treatment will become resistant in the person. All right? And they're going to, and now you have a bigger problem. Also, a person can contract this strain to begin with. They try these first-line drugs on them, rifampin, isoniazid, and they don't work. So you say, oh, you've got MDR-TB. Well, is there anything worse? Yes, there is. Okay, there is extensively drug resistance. Think extensive. Extensive is more than multi. That means they go by XDR-TB. These are strains that are resistant to at least 3 classes of antibiotics. So those 3 classes are isoniazid and rifampin. And now these guys have a third class that they're resistant to. Usually, but not only, but usually fluoroquinolones, because those are used usually on strains of Mycobacterium tuberculosis. And these are used as a second line. So now there's strains out there that are resistant to these 3 classes. So the question is, what do you do if a person has XDR-TB? Well, you go try different antibiotics, other classes. But you can see that, if you don't find the right one and it doesn't work, the person's going to end up dying. And that's why when it gets to this stage, often, there's a very high mortality rate with tuberculosis. Remember, that's one of the top 4 killers in the world. Okay, that's one of the reasons. All right.
So what can be done to inhibit the spread of antibiotic resistance? Number one, you cannot stop mutations. Okay? It's not like, oh, let's stop mutations. No, bacteria are constantly mutating. Okay? Spontaneous mutation, induced mutation. There's no way to stop that. You're always going to have bacteria, individual cells that arise that are resistant to some antibiotic. It always happens. So basically, you have to stop this part 2. You have to stop the selection part of it. You have to, or society has to prevent those resistant strains from being selected for, i.e., you've got to stop those strains from getting exposed to the antibiotic that's going to select them out. So, number one, use the antibiotics only when necessary, okay? Avoid the indiscriminate use, putting it in feeds, giving it at low levels prevention, giving it for a viral disease when you know that it has no effect on the viral disease. Also, good use, prevent its good use. Vaccinate people so they don't come down with the bacterial diseases like whooping cough, like tetanus. So if they're vaccinated, they shouldn't come down with those diseases. Therefore, you would not have to treat them with the antibiotic. So vaccination is good for many things. It also helps stop the spread of antibiotic resistance. With this idea of limiting the use, there are some antibiotics that are considered drug of last resort, not for the first reason we talked about. Okay, remember the first reason we talked about a drug being a drug of last resort is it has really bad side effects. So the side effects are so bad you don't want to use it. If there's antibiotics that are effective that have less side effects. But sometimes that's the only thing, so it's the last resort. Boom, use it. All right. Now the second reason something is a drug of last resort is that it's not their side effects. It's that medical science don't want them used very much. So they're purposely not used much. They're saying can only use it as a last resort when all the other ones don't work. Why? Because if we don't use it a lot, there won't be selection for resistant bacteria. And therefore, it'll decrease the chance that widespread resistance to that antibiotic will ever occur. So we won't use it a lot, and therefore there should not be the widespread resistance to this antibiotic among bacteria. So let's keep it as a last resort. Let's just keep it as a last resort. All right? So there's two reasons you can see drug of last resort. Another thing that can be done is actually to kind of stop the spread of widespread resistance and this is kind of interesting, is to use combination antibiotics. So I mentioned a Synarcid before. It's a streptogrammin. It's actually two streptogramins together. And each of them bind a different part of the 50S ribosome. So they have different sites. Okay? They're binding different sites. They're working as a team. Okay? And what they found is this, is that there's not many bacteria that are resistant to Synarcid. Okay, why? Well, there's two things in it. There's a pretty low probability that a single bacterium that will develop a resistance to two different antibiotics by mutation. So a bacterium having just spontaneously one mutation to lead to resistance to one of those streptogrammins, it's pretty rare, but it happens. But a bacterium having two mutations that make it resistant to both of those streptogrammin drugs that are found in Synarcid, very, 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 very rare. So using Synarcid then, let's say if a target bacterium is resistant to one of the antibiotics in there, it's going to get killed off by the other one. So therefore it won't become widespread because it's going to be killed off by the second antibiotic. All right, so there's your thing of Synarcid. It's got quinipristin and dalfopristin. Like I said, they are streptogrammins. All right, and it's kind of considered a drug of last resort too, because they don't want bacteria to become resistant to it. So they try to keep it only for situations where it's absolutely needed. Okay, because they want to decrease the chance of selection for it. This is the final slide of the day. First of all, we have a link to a video here. It's only about 5 minutes long. It's supported by the Bill Gates Foundation, and it summarizes a lot of the concepts we talked about with antibiotic resistance. It's called antibiotic apocalypse. So it's worth 5 minutes watch. It's a good summary. Secondly is a chart that comes from the online book. On the y-axis, you have antimicrobial approvals. That's new antibiotics over these little 5-year periods for the last 30 years. And you can see the number of antibiotics being approved has going down. And you must be thinking to yourself, hold it, if antibiotic resistance is getting worse and worse, how can it be that pharmaceutical companies are putting out less new antibiotics? Doesn't make sense. Well, you have to understand it's a financial matter. Little cocktail. A pharmaceutical company spends about 10 years and $300 million to bring one antibiotic to market. That's a big investment. And then they have to sell it and make enough money and profit for their shareholders. So they got to recoup the $300 million, plus they have to make a lot after that. If an antibiotic comes to market and suddenly there's a lot of widespread resistance to that antibiotic, then that antibiotic is not going to be used much. It's not going to be sold, and that pharmaceutical company is not going to make that much money. And that's been happening a lot. So some pharmaceutical companies are shying away from making antibiotics for that reason. And that's why new ones are not being developed at the rate they should be. Now, recognizing this problem, the United States government is trying to set up new ways of reimbursing, paying, encouraging pharmaceutical companies to take the risk to go forward with antibiotic development. But it's something that's being worked on right now, and hopefully it's resolved, because we can't keep up with this. This number has got to go up. If not, we humanity are going to be in serious trouble in our battle against pathogenic bacteria. That concludes today. Thank you very much. Take care. Stay safe. Try not to get sick. See you in the next lecture.